Yeah, and Henry, I will hand over to you to introduce
Hey,
yourself and Claudia, of course.
thank you very much, Tatiana. So my name is Henry Valentin, and it's my pleasure today to moderate this session. Maybe a few words about myself. So I'm located here in Chesterfield, Missouri, at the Chesterfield site of Bayer. As already mentioned, I'm a Bayer Science Fellow. And originally, as you may be able to hear on my accent, originally I grew up in Germany. I studied microbiology in Göttingen and have been with Bayer or its precursors since 1996. I've worked on a variety of different projects around metabolic engineering. Lots of them were associated with lipids, and so I have some particular interest in this presentation because of that. So maybe a few words about Claudia. Claudia is a group leader at the Institute of Supramolecular Science and Engineering at the University of Strasbourg, France. She obtained her PhD in biomolecular science at the University of Trento, Italy, focused on the synthesis and activity of primitive catalysis. Later, she moved to the MRC Laboratory of Molecular Biology in Cambridge, UK, as an MSCA fellow, and then to the Department of Chemistry at the University of Cambridge as an 1851 research fellow, where she tackled fundamental questions related to the emergence of functional primitive cells. Currently, her group focuses on prebiotic supramolecular chemistry and the self-assembly of primitive lipids into functional primitive cells that are able to replicate. And with that, I'd like to hand over to Claudia to tell us a little bit more about that.
Thank you very much. So I will first share my screen just to be sure that everything works fine, and then I will start. So hopefully everyone can see my screen. So thank you very much for the introduction. Thank you very much for inviting me and for giving me the chance to share my research. So we'll talk about my research in a few slides, but I figured that I could expand a bit on the path that led me here, just because I feel like there are a few things that were absolutely key for me in my career path, and that most likely are the reasons why I'm here today. So I decided to create a map of my, let's say, path in science. So as you can probably already imagine from my name and also probably from my accent, I'm Italian. I did my PhD, as it was said, at the University of Trento, even though I'm from Tuscany. I'm from a small city called Siena that is close to Florence, where I did my bachelor, and then I moved for my PhD to Trento. And as it was just said, I started working on the big problem of the origin of life already as a PhD student. And my PhD supervisor is American. He married an Italian scientist, which drove him to set his lab in Italy rather than stay in the US. And this was one of the first times that I had the chance to experience people from different culture, coming from different backgrounds, because the way the lab was set was very much in the American style, and we had people from all over the world. This also gave me the opportunity to be open to opportunities to spend time abroad during my PhD. And so during my PhD, I spent time both in both Cambridge, in the UK, and in the US for about 3 and 4 months. I learned that I loved spending time abroad, and I loved meeting different people from different backgrounds and with different expertise. And so when I was looking for a postdoc position, I decided to move away from Italy and join the same group that I had joined during my PhD as a visiting PhD student in Cambridge, which thanks to a fellowship, I got the chance to start becoming a bit more independent, but I still kept working on origin of life research because I was really interested in that. And when I was in Cambridge also, I had a chance to spend some time abroad. I spent some time in the US again, until a couple of years ago when I started my independent lab in Strasbourg. And the reason I spent a few minutes talking about what drove me here is because I think there are three key aspects that were absolutely fundamental in allowing me to become the group leader that I am right now and the scientist that I am right now. The first one is, as I said, the possibility to meet people from a lot of different places around the world and doing a lot of different types of research. So the experience, the possibility to experience different cultures is something that has been really important for the development of myself as a scientist, but also as a person. The second aspect that has been absolutely key for me has been networking and mentoring. So meeting people at conferences within the department, during Zoom calls and webinars, people that you would not otherwise meet and have the chance to exchange ideas, for me has been absolutely key. And the last part is, as you will see most likely from my presentation, I have a chemistry degree. I have a PhD in biomolecular sciences. I do a bit of everything. I don't feel I'm an expert on anything specifically, but I love the idea of combining a lot of different disciplines. And so my presentation hopefully will show you a bit of chemistry, will show you a bit of biophysics and a bit of biochemistry. So these are the key aspects that I think I've experienced during my career path and that are at the basis of what I'm doing today. So, having spent already enough time on this, I will go back to the title slide. And as you can imagine from the title and probably from the introduction, what we're going to talk about today is early life, intended as the life that emerged about 4 billion years ago on our planet. Now, you can already imagine that we don't have that it's difficult to be sure about what happened 4 billion years ago. We don't have witnesses about what happened. We don't have a time machine to go back in time and figure out exactly what happened. So what we are doing to understand how life might have started is combining information that we can get from astronomers, from earth scientists, from physicists, from chemists, and from biologists, looking at how life is today, and try to combine all of this information and understand what kind of primitive life forms could have emerged on early Earth, where, how, when. So the motivation behind what we are doing comes from the life that we have nowadays. And when we think about modern life, modern cells, we think about, from a chemical point of view, systems that can be considered as built by three key ingredients. So if we think about a living cell, we can reduce a living cell to a mixture of three ingredients: nucleic acids that are key to store and transfer the genetic information, proteins that are key to power the cell from an energetical point of view, and then membranes, which are the container that separates the inside from the outside. And when one looks at the molecular aspects of all of these building blocks, there are a lot of questions that we still don't know, for which we still don't have an answer. And I'll just give you a couple of examples. The first example is, we know that nucleic acids, DNA, RNA, are made of small monomers that are called nucleotides. And there are 4 or 8 nucleotides that combined together can give rise to DNA and RNA. When it comes to proteins, there are around 20 amino acids that we can combine to build up a large variety of proteins.
Claudia,
Instead, when it...
are you advancing the slides? Because we see
I am on a
still...
slide that says, why is life the way it is? Is that right?
Oh, okay. So we are still on slide 1. Okay, sorry. Thanks.
Yes. Yeah. No, it's totally fine. Absolutely. Feel free to interrupt whenever you want. So we have a relatively small number of monomers, of nucleotides to build nucleic acids. We have a relatively small number of monomers, amino acids, to build proteins. When it comes to lipid membranes, there are more than 100 different types of lipids that can be combined to build membranes. Why do we need such small alphabet of nucleotides and amino acids and such a large alphabet of lipids to build cells? We don't know. Second question I would like to put your attention on is relates to stereochemistry. When we think about nucleic acids, DNA, RNA, we know that nucleic acids are built with sugar that have one specific stereochemistry. These are these sugars. When it comes to proteins, we know that proteins are made of amino acids that have one specific stereochemistry. This is L-amino acids. When it comes to lipids, and we look at all living systems, we have both stereochemistry present in living systems. So why do we have only one stereochemistry for amino acids, proteins, and nucleic acids, but we have both for lipids? These are questions, just examples of questions for which we have absolutely no answer yet. So now I am advancing my slides. The common approach to try to answer these questions and understand how life might have started is basically to first understand which were the ingredients that could have been available on early Earth, so synthesizing the ingredients of life. And this is something that I've done during my PhD and during my postdoc to some extent for all the different biological building blocks. And the next step is combining all of these ingredients and trying to understand how living cells could have arose from them. So to build a living cell, what we tend to do is look at what we know that works, which is a modern cell. And as I was mentioning earlier, we can imagine a modern cell as a sort of combination of three key ingredients: nucleic acids, proteins, and membranes. And when we think about how a primitive cell could have looked like, we think about a cell that is made basically exactly of the same ingredients, just in their primitive versions. Primitive meaning more simple from a chemical point of view and also in terms of their properties and functionalities. Now, what my lab is focusing on at the moment is one of these three key ingredients, which is the membrane and the molecules that can form this membrane. And so the key question that we're trying to address in the lab is what kind of membranes, what kind of compartments could have formed and could have evolved on early Earth, leading us to the membranes that we have nowadays in our living systems. And so this big question can be decomposed in three different parts. I'm going to tell you a bit about everything what we are doing in the lab. So the first point is what kind of molecules can be combined together to build a primitive membrane. So how we can combine from a chemical point of view different parts of molecules to build lipids. What kind of function these lipids, once assembled into a lipid membrane, could have. We are thinking about transport, we're thinking about catalysis. We're also just thinking about what's the effect of other molecules on the properties of these membranes. And then ultimately, we would like to end up with a system that behaves exactly as modern cells do. And when we think about modern cells, we think about a system that can grow and divide. And this is ultimately the goal we would like to achieve for primitive cells as well. And so I just thought that I would show you the faces of the people that are actually working in the lab, because what I'm going to tell you is not what I've done in the lab, but what they've done in the past few years. And so I will start from the first challenge, which is understanding what kind of molecules could have been available on early Earth. So again, we're talking about 4 billion years ago, roughly. We're talking about Earth, a planet that was very different compared to the planet we have today. One very simple difference is the amount of UV light that would hit the surface. Today, we are used to think about UV light coming from the sun being to some extent filtered by the ozone layer. 4 billion years ago, there was no ozone layer. There was much less oxygen in the atmosphere. The amount of UV light that would hit the surface was much higher and much more intense. So just to give you an idea of what kind of differences we're thinking about when it comes to the surface of the Earth, which is where the chemistry we're going to talk about could have taken place. So for the first question, is what kind of molecules could have formed on early Earth that would have been able to assemble into lipid membranes, the same kind of lipid membranes that form the boundary of our cells nowadays. We know that one of the key components of modern cells, of modern cell membranes, are called phospholipids. And so what we were interested in looking at was what kind of phospholipids could have formed on early Earth. And assuming that multiple phospholipids, even phospholipids that are not natural, could have formed, how the right ones could have been selected. Now, for those of you who are not familiar with what phospholipids, what's the chemical structure of phospholipids, I decided to draw it here. So you can see that phospholipids are molecules that have a hydrophobic part, the black part on the molecule, and a hydrophilic head group, which is polar, which is represented in purple. Combining a lot of these molecules forms a lipid bilayer that is what we call a lipid membrane. So the first, if we look at, if we get inspired by biology nowadays, the biosynthetic pathway to synthesize this kind of molecules in our cells start from the synthesis of a precursor that is called phosphatidic acid. So this is what modern cells do. They take a molecule that is called dihydroxyacetone that you can see on the left side of the screen. This molecule gets phosphorylated and reduced. You end up with a glycerol phosphate molecule, that is the one you see in the center of the screen. And this molecule is still very hydrophilic, so it's perfectly soluble in water. But as soon as you acylate this molecule, so you add some lipid tails, some hydrophobic tails, you end up with a molecule that is called phosphatidic acid that is capable of self-assembling into lipid membranes. And that is the precursor for more complex phospholipids. Now, this is what happens in our cells thanks to a lot of enzymes that take care of all of these transformations. So the question is, 4 billion years ago, when we did not have any of these enzymes, how could we envision all of these chemical transformation to take place? So what we would need to have is ideally a series of transformation very similar to what we have nowadays in our cells. And so luckily enough, there were already groups that had started to look into this problem. And so my postdoc supervisor in Cambridge had previously looked at the possibility in the absence of enzymes to transform dihydroxyacetone, so the precursor for these molecules, for these phosphatidic acids into glycerol phosphates. And you can see that you can already see a difference compared to what I've shown you before, because when you start you don't use any more enzymes, but you start using more, let's say, simple chemicals, you start losing, for example, in selectivity. And so you might end up with a larger amount of species of products. So in this case, reduction and phosphorylation of dihydroxyacetone leads to a library of glycerol phosphates. And when I was a postdoc, what I did was trying to convert these glycerol phosphates into phosphatidic acids. But what you might already be noticing is that I did take one of these glycerol phosphate and convert it into phosphatidic acid, But unfortunately, the one that I worked with is a non-natural lipid. So earlier I told you that there are hundreds of different lipids in living systems. Unfortunately, this is one of those that is not present in living systems. And so this poses a question, which is, if on early Earth we have such a messy system where we have so many different substrates, how can we select for the one that we need to form natural phospholipids and get rid of all of those substrates that we don't want because they lead to non-biologically relevant molecules? So this is the question that we tried to answer in the lab in the last 6 months or so. So this is work of David and KK in the lab. And the first thing that we wanted to do was to understand if we can take the natural glycerol phosphate and convert it into a lipid. And the answer is yes. So if we take glycerol phosphate, glycerol 3-phosphate, we use relatively mild conditions to add the lipid tail. You end up with a mixture of molecules that have 1 or 2 lipid tails and that have behaviors, the same behavior that a lipid molecule would have. This is a phosphorus NMR that shows that the reaction works. But what you can see is that the peak corresponding to the substrate, that is the one on the left, the black one, is very abundant compared to the small peaks, colored peaks of the product. This means that the reaction is not as efficient as we would like it to be. On the contrary, if we mix together the natural and the non-natural substrates, and we expect to form lipids from both of these substrates, what we end up with is a mixture, is a solution that contains a mixture of molecules. But you can see that the peak on the right, which is the pink one, is the most abundant peak, and this is the non-natural lipid that we don't want. So the problem here is that if we start from a mixture of substrates and only one of them could lead to biological lipids and the other one doesn't, but the one that doesn't is the one that reacts more efficiently, then how is it possible that on early Earth, where you couldn't use HPLC or other kind of instruments to select for the molecule that you want, how could we select for the natural phosphatidic acid over the non-natural one? So what we decided to look into was what this molecule do once they are put in water. And the first thing that we've noticed is that if you put these two molecules, if you separate these two molecules and you put them in water, they do assemble into lipid membranes. So both of them can form lipid membranes, but the question we had was whether these lipid membranes could form in different conditions. And so if it was the conditions that we use that can select for the natural phospholipid compared to the other. So what we decided to do was to see what's the minimum concentration that is needed for these two molecules to self-assemble into lipid membranes. And the way we did that was with an assay that is called myrocyanin 540 assay. Basically, this is a molecule that intercalates into the lipid bilayer and tells you when you have lipid membranes or not. And by looking at the UV spectrum that you get of this molecule, you can understand when or not you have liposomes. So the results that you're seeing in the graph in the center of the slide right now is the outcome of this assay that tells you what's the critical vesicle concentration, which is the minimum concentration you need of a lipid to form liposomes. And in this case, we have 0.7 millimolar critical vesicle concentration for the natural phosphatidic acid. And what we were very pleased to see is that when you do the same experiment with the non-natural phosphatidic acid, the critical vesicle concentration is higher. This means that even though the non-natural phospholipid forms more efficiently, you need to make much more of it in order to self-assemble into lipid membranes. So that made us, led us to think that the natural phosphatidic acid could be selected at the supramolecular level, not at the molecular level. We confirmed that by looking at phospholipids, phosphatidic acids that have different chain lengths. So this happens, this is consistent and happens regardless of the chain length. And so the take-home message from this part of the study is that maybe not at the molecular level, but it's the self-assembly that could lead to the selection of the natural, the biologically relevant phospholipids over the non-biological ones. So let's assume that I have convinced you that on early Earth, we could have formed the precursors of modern phospholipids. How do we go from these precursors to the modern phospholipids, the complex phospholipids that we have in modern membranes? So once again, let's have a look at what biology does. Biology takes phosphatidic acids, activate the phosphatidic acid, and then the phosphatidic acid undergoes nucleophilic attack. The activated phosphatidic acid undergoes nucleophilic attack, and then you end up with a molecule that has something attached to the phosphate group. And the interesting thing is that even though it might seem a very small change in terms of the molecular structure, that small purple thing, that purple moiety that is attached to the phosphate changes completely the function of the phospholipid. When we look, you might have heard of phosphatidylcholins. Phosphatidylcholins are the major components of eukaryotic cell membranes. They are responsible for the structure of the membrane. But when instead we have phosphatidylethylamines, they are key in in functions related to protein binding or in modifying the curvature of the membrane. And in the case of phosphatidylserine, it plays a key role in changing the charge of the membrane. So even a small variation in the functional groups of the head group of the phospholipids could have a key role in the function of the membrane itself. So what we were interested in was to understand if we could unlock a chemistry that could lead to the synthesis of these molecules. And so once again, we had to try to find ways to do that without any enzyme. So the first thing we did was to find a way to activate the phosphatidic acids. And we found that a good way to activate the phosphatidic acid was to convert them into their cyclic analogs. So if you form a cyclic glycerol phosphate moiety that you can see here in the middle of the screen, the question then becomes, can you expose that to a nucleophile and synthesize the same kind of phospholipids that we have in our cells today? So I'll just, spoiler alert, I'll just tell you that the answer is yes to this question. And this is what Fidan and Maya have been working on in the past few years. So if you just imagine to mix together the ingredients, nothing really happens. So in this case, we were trying to to synthesize the head group of phosphatidylcholins, but if you just mix them together, nothing really happens. However, if you give them a bit of heat, so a bit of energy, under relatively mild conditions and in the presence of an amine catalyst, what happens is that the two molecules merge together and we ended up with the head group of phosphatidylcholins. The same happens if you repeat the same reaction in the presence of serine, in the presence of ethanolamine. And these, just to remind you, are the key ingredients, the key head groups that unlock a large variety of functions in modern membranes. But this could be extended also to a lot of different other types of nucleophiles. And this is something that I think is very interesting, not only because it allows us to expand the library of lipids that could have been available on early Earth, but this also gives us the opportunity to synthesize completely artificial lipids that are not present in modern membranes, but can unlock completely new functions for lipid membranes. Something that I want to put focus on for a moment is that you might have noticed that we have in this phosphorus NMR we have two peaks related to the products of our reaction, and the reason is that the glycerol cyclic phosphate substrate that you can see here on the left can be opened in two different points, and so you end up once again with one of the isomers that is the natural, the biologically relevant one, and one that is not biologically relevant. So once again, we are creating a problem because we need to find also a way to select the natural lipid head group over the non-natural one. In this case, it was relatively simple to find a solution because if you just follow the reaction over time, you can see that the peak on the right that corresponds to the non-natural head group decreases over time, so it gets degraded over time, while the natural head group peak sticks around, which means that this is just a representation of the same data in a different way. This means that we can accumulate for much longer the natural head group of modern phospholipids and get rid much more easily of the non-natural head group of the phospholipids. We have repeated these experiments also with lipids, not just with the head groups, and the outcome is that we've identified what we think is the first regioselective pathway that could have led to natural phospholipids under primitive conditions. And this, just to do some self-advertisement, was just published actually last week in JAX, and so this is one of the first outputs of our new lab in Strasbourg. Now, I've told you a bit about the what kind of molecules could have formed on early Earth and how they can assemble into lipid membranes. I'll just give you just a feeling of what else we are doing once we have formed all of these membranes. So the first thing that we are interested in understanding is how these membranes behave when you change the conditions, the environmental conditions around them. And so the first thing that we were interested in was understanding how membranes primitively respond to temperature. And so what I want to show you here is a short video. So in this case, we've looked at very simple lipids. You can see the structure in the corner of the slide. This is myristoleic acid. It's a molecule that self-assembles into membranes, as you can see from this microscopy image. And what hopefully you will see now is a movie where we have taken lipid membranes that are fluorescently labeled. The only thing we are doing is increasing the temperature of the solution. And what you can hopefully see now is that the morphology of these structures changes completely. So now we no longer have lipid membranes, but what you are seeing on the screen at high temperature are oil droplets. So what happens is that the lipid membranes, when you expose them to high temperature, collapse and form oil droplets that, as we know, tend to coalesce with one another. The question then became, is this reversible? The answer is yes. So what you can see is that from the surface of the oil droplets, when we decrease the temperature, new lipid membranes bud out and form. So this was something very interesting because we were not expecting such a easy conversion from lipid membranes to oil droplets and vice versa. This is something that we've followed by microscopy, as you've seen from the movie, but that can also be followed with other tools. We use UV spectroscopy and fluorescence spectroscopy. So the gray line that you see here is just following the turbidity of the solution. We know that oil droplets give much more turbid solutions and so scatter the light much more compared to lipid membranes. You can do the same by using fluorophores, embedding them into the membrane, and you can see that there's a very sharp transition at a certain temperature due to the fact that the lipid membranes collapse into the oil droplets. So something that we are currently looking at in the lab is whether the same behavior can be induced also by the presence of some additional biomolecules in solution. So this is something that Nina is looking at, the effect of amino acids on lipid membranes. And we can see that the temperature at which the collapse occurs changes quite drastically when we go, for example, from glycine, which is a very neutral amino acid, to arginine and lysine, here in red. You can see that the temperature changes, this collapse happens at almost 30 degrees higher temperature compared to when you don't have positively charged amino acids. And this is allowing us also to measure the dissociation constant, so the affinity between these amino acids and liposomes. That overall tells us that there's a strong interaction between amino acids and lipids, and they modulate the stability of these membranes. Now, the last thing I wanted to tell you a bit about is something for which we have only preliminary data, but that I think is taking us to the next step, trying to get much more closer to the kind of functionality that modern membranes have. And so, as I was mentioning earlier, one of the key features of cells is that they can grow and divide. And so what we were curious about was whether primitive cells could do exactly the same. And there are actually some bacteria that are called L-form bacteria that divide according to the biophysical method or way, which means that they don't use complex biological machinery to divide, but what they do is they overproduce lipids. So the membrane gets deformed. And once the membrane is deformed, this leads to spontaneous vesiculation and budding and division. So the question we had was, we definitely didn't have complex biological machinery on early Earth in primitive cells. Is it possible that we can induce a deformation of the membrane in the same way this happens in L-form bacteria and achieve growth and division of primitive cells? So the way we decided to go about this was to study the possibility to deform lipid membranes in very simple ways, and to generate a fission plane that would allow, under certain environmental conditions, to actually detach and divide the lipid membrane. So this is something that Johan has been looking at, trying to find ways to induce the presence of a fission plane on the membrane, so a specific point on a lipid membrane that would enable the separation and the breakage of the break of the lipid membrane. And this is something that some of you might have read or seen before. Sometimes when you mix together lipids on a membrane, they are not homogeneously distributed, but they form domains that are enriched in one lipid or another. So what you are seeing in these microscopy images are some black dots on a much lighter domain, let's say, and the black dots are enriched in one lipid. The lighter domains around them are enriched in a different lipid. Now, this is something that happens with modern phospholipids, but we are trying to achieve the same thing with much simpler phospholipids that could have been present on early Earth. The second thing we are doing is we're trying to induce the formation of the membrane, as I was saying before, so that then we can combine the two things and try to achieve division of the membranes. And I just wanted to show you something that I find very exciting, because we've seen that we are starting to see, by just playing with the temperature in our system, we are starting to see a lipid membrane that starts to get deformed and starts to get towards that point in which, under certain environmental conditions, for example, inducing some osmotic stress, they could potentially divide at the fission plane. With this, I will wrap up, summarizing what we've seen so far. So we've seen that some of the molecules that could have formed on early Earth could have self-assembled into lipid membranes similar to the ones we have today. These lipid membranes respond to their environment by converting into different structures or getting more or less stabilized by interactions with other biomolecules. And ultimately, they can deform up to a point in which they can undergo growth and hopefully division in the same way that modern cells do. So where can we go from there? We have seen that we can synthesize a large variety of lipids under primitive conditions. But as I was mentioning, the chemistry that we are discovering could allow us to synthesize many more lipids without restricting ourselves to the lipids that we find in modern membranes. We could synthesize lipids that are completely artificial, and this could give us the opportunity to build liposomes and lipid membranes that have completely unexplored properties. We are starting to understand what's the effect that other biomolecules have on lipid membranes. And this could give us the opportunity to design functions in lipid membranes when it comes to transport, it comes to signaling, it comes to the properties in terms of permeability and stability of the lipid membranes. So this is also something very exciting that we are exploring. And then finally, we are building lipid membranes that are made of so many different lipids that could start showing the functions that modern membranes have. For example, their ability to grow and divide in the same way that cells do today. With this, I just have one last slide of acknowledgement. These are most of the people that have contributed to the science that I've shown you today, and that are contributing to a lot of other projects that we have running in the lab at the moment. Thank you very much again for inviting me and for hosting me, and I would be very happy to answer any question you might have.